Hi there, science fans. Welcome to another episode of Science and Other Stuff for Senior Citizens. My name is Dan Casciano. The theme of the channel is to describe science in terms that would be understandable to all. If you like these videos, please subscribe and ring the bell to be informed of new videos I publish. Don't forget to share. In this video, I will describe a few of the ways the discipline of nanotechnology impacts the development of COVID-19 vaccines, and also some ongoing laboratory research that will impact development of future virus vaccines and therapies. So what is nanotechnology? The scientific discipline of nanotechnology is the field of research and innovation conducted at the scale of atoms and molecules. This nanoscale includes structures ranging from 1 nanometer to 100 nanometers. 1 nanometer is 1 billionth of a meter. As can be seen in the diagram on the left, many scientific areas impact the field of nanotechnology. Much of the innovation comes from the physical sciences, but as can be seen in the diagram on the right, various fields in the biological sciences are collaborating with the physical sciences in many ways, especially in the development of another new area of research and application referred to as nanomedicine. Nanomedicine encompasses the biological applications presented in the red rectangle. The remainder of this video will concentrate on drug delivery, or in this case, vaccine delivery. These diagrams indicate some of the nanoparticles used in the field of nanomedicine. They range from inorganic particles like iron and silica, to particles conjugated or attached to proteins, drugs, antibodies, and various polymers. Some of these molecules that are attached to these various drug conjugates allow the targeting of the drug to a specific cell or organ. For instance, if a cancer drug was attached to a molecule that would only target a lung cancer cell, then the normal surrounding cells and tissues in the lung and elsewhere would escape the lethal effects of that drug. At present, most of the cancer drugs in use do not discriminate between normal and cancer tissue. If there is interest in the use of targeted cancer therapy, leave a comment in the comment section and I will develop a video describing that technology. However, the remainder of this video will describe lipid-based nanocarriers and viral nanoparticles as they relate to COVID-19 and some innovative developments occurring in the laboratory. Use of a lipid nanoparticle as a carrier for the COVID-19 viral gene that codes or specifies the spike protein is described in this slide. The nanoparticle is a non-targeting particle indicating that it is taken up by all cells in the human body that it comes in contact. Some of these cells will be immune cells. If a targeting nanoparticle was available, it would be taken up mainly by immune cells. This type of messenger RNA vaccine was initially developed in a laboratory by scientists who isolated the RNA genome of the COVID-19 virus and determined the gene that codes or specifies the spike protein of that virus. The messenger RNA from that gene is mass-produced and then inserted into the carrier lipid nanoparticle prior to injection into a human. After the lipid nanoparticle enters into specific cells, the spike protein is synthesized in the human cell and excreted into the bloodstream that is then recognized as foreign by our immune cells. This recognition results in the recruitment of a variety of immune cells that then produce antibodies to that spike protein that are used to inactivate a COVID-19 virus. In addition to being recognized by the cells that make antibodies, another cell type called T memory cells are developed that remember the structure of the spike protein so that a much more rapid immune response can be made by the human upon future invasions by the COVID-19 viruses. This slide depicts the same mechanism as found in the previous slide, except that the nanocarrier is an inactivated common cold virus called adenovirus. The SARS-CoV-2 spike protein gene is isolated and inserted into a portion of the adenovirus genome, resulting in an engineered genome that is then inserted into the adenovirus vector or carrier particle. This nanoparticle is then injected into humans and the engineered genome is translated into the SARS-CoV-2 spike protein, which is then recognized by the human's immune system as foreign, resulting in the production of antibodies and T memory cells. Now that we have several vaccines against COVID-19, questions arise whether these vaccines will work on mutants or variants of this virus, and if these vaccines will protect us from other strains of the coronavirus. Research recently published in the journal Science investigated the potential to train the immune system to produce antibodies to multiple strains of coronaviruses with a single injection of a vaccine. The research is based on a technology called Mosaic Vaccine Platform that is represented visually in the cartoon on the left. The central cage-like nanoparticle is attached to its several different spike protein fragments or antigens called antigen A through antigen J. When a system like this was injected into mice, a model system used for work in immunology, there was a diverse antibody response such as that depicted in the cartoon on the right side of the slide. The initial results are encouraging because the mice began producing antibodies that could neutralize many different strains of coronavirus. While studies like this are still in their infancy, the potential for application in the human is enormous. This slide depicts another future use of nanoparticles as nanodecoys against COVID-19 as well as other viruses. In diagram A, 2 nanodecoys are being produced, one against the human cell's receptor that allows entry of the virus into the cell, and a second that helps in regulating the cytokine storm that may be generated in severe infections of COVID-19. The receptor is the ACE2 receptor, and the cytokines requiring regulation are IL-6 and GM-CSF. In diagram B, the nanodecoy competes for the spike protein located on the surface of the virus, resulting in the patient experiencing a lower virus load. In diagram C, the nanodecoy competes for the cytokines, thus reducing their concentration in the blood, resulting in decrease in severity of an infection. This diagram depicts research ongoing in nanomedicine laboratories. A variety of nanoparticles are being developed that are decorated with a variety of targeting molecules. In the case of vaccines, the nanoparticles contain vaccines that are targeted to the cells of the human immune system, allowing more efficient delivery of vaccines, resulting in a more rapid response to the invading organisms. More about these targeting nanoparticles in upcoming videos. Although vaccines are beginning to be administered to humans, we must continue to clean or wash our hands often, avoid close contact with sick people, practice distancing 6 to 10 feet, avoid indoor and outdoor crowds, and wear a mask properly. We will continue following these CDC guidelines until we reach herd immunity. For more information about science, visit my website at www.dcasciano.com. If you have any questions or would like me to develop a video on a specific science subject, leave them in the comments section below or email me at dan@dcasciano.com. Be sure to like this video and subscribe to my channel and ring the bell so you can be informed when I upload a new video. Thank you for watching Science and Other Stuff for Senior Citizens. Bye.